this speaker series, I've been calling it unofficially Celebrating Wonder, because, I mean, that's really what it's about, science. All of our wonderful speakers have dedicated their lives to scientific inquiry. And I mean, I have to say that my friends who are scientists are the hardest workers. They work for no pay and practically no recognition, and they do this incredibly important and fundamental work, helping us answer really important questions about our world. And I'm really impressed with every speaker we've had, and I'm really excited and impressed with Dr. Shier's work. We're so delighted to have her speak to us today. If I told you everything about her work, that would fill the entire hour. So I'm just going to keep this short and just say she is an expert in her field. She's conducted important conservation research for over 20 years. At the moment, she conducts research at the San Diego Institute for Conservation Research as the Brown Endowed Associate Director of Recovery Ecology. She's worked with diverse species, including kangaroo rats. She has a lot of them up here. Kangaroo rats, ground squirrels, pocket mice, black-tailed prairie dogs, ringtails, Tasmanian devils, yellow-legged tree frogs, and so many more. She's going to share with us about her experience of these fascinating animals and her insights on conservation strategies. If you have any burning desires, burning questions, save them till the end. She'll be doing a question and answer period. And I am so delighted to introduce the illustrious Dr. Debra Shier.
Wow. Thank you, Julie. So yeah, as Julie said, thank you all for coming, by the way. It's fun for me to be here. We've lived in the canyon for 17 years, and it's fun to share some of my research with all of you. I chose a starting slide to just sort of represent the variety of species that I've worked with over the years. But I'm going to today focus on some of the local species we have here in Southern California. At the end, if you have questions about some of the other species, I'd be happy to talk about those as well. So we all know that we're in sort of an extinction crisis, that humans are driving extinction through habitat fragmentation and habitat destruction, and that a lot of animals are being driven out of their homes. And so in order to prevent extinction in these vulnerable species, wildlife managers use relocations. So we're trying to move them from areas where they're being impacted to areas where they can be protected, in national parks and private conservation land, or in reserves here in the United States. Now, the goal of relocating animals is obviously to get them to survive where you want them to live, for them to reproduce, and for you to establish a viable population. But often, we don't reach those goals. It's really tricky to reintroduce animals and have them make it. So they might initially just disperse. They might disperse from you put them somewhere and they don't want to stay. And they may disperse because they're site faithful. They might not be familiar with the animals that you put them out with, and so they want to leave. Or they might not be familiar with the physical characteristics of the environment that you put them in. And when they run away from those release sites, they're often extremely vulnerable to predators. So those first few weeks to months after you translocate them or you reintroduce them from captivity are very risky for released animals. And so what we try to do is we try to improve those methods so that we can actually successfully move animals. So I'm going to talk a lot about that today with a variety of different species. Now, wildlife managers try to prevent animals from leaving by putting animals into acclimation cages. So if you hold them on a site for a short period of time, it's thought that you can dampen dispersal. So that means for you guys out there that they won't run away. But often, even if you do that, the released animals will leave the release sites and they're more vulnerable to predators. So what I do is I study basic behavioral ecology. So each animal behaves differently, and they're adapted to their environment and all the in their ecosystem, all of the animals that they live with. And so if we know something about their behavioral ecology, we can sometimes use that information to improve our methods for relocation. So these are the kinds of things I'm looking at. What kinds of groups to move, what kind of habitat that they might select so that we can look for that at the release site. Do they have appropriate anti-predator skills if we're talking about moving a prey species? What kind of individual variation they have in their temperaments and how that might affect our release success, and using scent and acoustic communication to try to dampen dispersal. So I'm going to talk about some of those things today. I'm going to start with one of the species I love the most, the Stevens kangaroo rat, and I'm going to talk about these guys because I've done an extensive amount of research on them with regard to their behavioral ecology and relocation strategies. Now, what do you guys know about heteromyids? They're probably not a lot, because they're a nocturnal species, or they're a family of species that are nocturnal, and they're pretty small. So most people don't even know that they exist. Because we're typically not out active at night. But heteromyids are kangaroo rats, kangaroo mice, and pocket mice. And I love to show this slide because people think, why are you trying to save a rat? And truth be told, kangaroo rats are not like other mice and rodents that you find running through your kitchen cupboard, right? They are no more closely related to those mice and rats than we are to monkeys. So they are really interesting species, and they're critical for ecosystem processes in the areas where they live. So they're found mostly in the western United States and down into Mexico. They are granivores. Does anybody know what that means, granivores, besides my children? They are seed eaters. And so they eat seeds almost exclusively. And one of the coolest things about these species are that they are adapted for living in our dry environments that we have in Southern California. And everything about them, their behavior, their morphology, their physiology is adapted for living in these dry environments. In fact, they never have to take a drink of water, ever in their life. They are, wouldn't that be great in the face of a drought if we could figure that one out for ourselves? Everything about them. So they have external cheek pouches, which means that they put their seeds outside of their mouth in their pouches when they cache them to bring them back to their burrows. And they do that because if you put seeds in your mouth, you're removing water from your mouth. Their kidneys are specialized so that they basically excrete almost no urine. They have low frequency hearing that allows them to detect the silent sound of a barn owl's wing beat. And they drum their feet to talk to each other. They are super cool animals. So in addition to just having all these amazing adaptations, they are critical for ecosystem function. So there was a really nice study that was published in Science in 1999 that showed if you remove the kangaroo rats from a desert scrub habitat, in 5 years, it changes completely. It type converts to a desert grassland. So they're really, really important for maintaining the native species of plants in their area. They are primary prey for a variety of animals. They're like popcorn out there. Everything wants to eat them. So if you get rid of the small rodents, you're actually, losing a prey base. They also dig burrows, which increases soil hydrology and nutrient cycling. So they're cycling nutrients out there because they dig. Yeah? So they're really important. This is the home range of the Stevens kangaroo rat. It's extremely small. It's down in Riverside and San Diego counties, and this was actually the range in the 1980s. We have actually lost half of this habitat due to development since then. Half of the land was promised to be conserved in reserves, and the other half was given up to development. So part of the problem is the reserves were not designed for kangaroo rats. And so if you think about what our habitat looks like in California, we think of grasslands on all of our slopes. But this is non-native grass. This is oats and bromes, which create basically a carpet that covers the landscape. And kangaroo rats, they not only drum to talk to each other, they need the surface of the substrate to be open so that they can sandbathe, which helps them keep the oils off of their fur, but also allows them to communicate territorial ownership to their neighbors, because not only are they nocturnal, they're solitary. So they live alone, and they need to coordinate mating and, you know, I live here, you live there, with their neighbors. And so this is a problem for them. So development continues. Our reserves often contain unsuitable habitat for the species, and previous translocations for kangaroo rats before we got involved have failed. In fact, one biologist moved 600 kangaroo rats in a translocation effort, and not a single one lived to 11 months post-release. So we had a lot of work to do. It was sort of a doom and gloom situation for the kangaroo rat, but we came in, my team came in, and we took on the challenge of trying to figure out how to incorporate the species behavioral ecology in translocations to see if we could improve our release outcomes. So this is what suitable habitat actually looks like. This is a native grassland. You can see the bunch grasses growing up, and you can see the open areas in between, and this is a kangaroo rat burrow. So they use those open areas, like I said, to sandbathe, and they also use them just to move through the landscape. We have very little of this. Let's see. So we did our work on the Southwestern Riverside County Multi-Species Reserve in Temecula, California. There's a fairly extensive system of reserves in Riverside County if you haven't had a chance to go down and visit it. And they do a pretty good job with management down there. And I just want to start with one of the experiments that I ran to see basically who to move together when we do a translocation. So as I mentioned, they're solitary. So you might think that you could just pick any kangaroo rat up and trap however many you need and move them. But from behavioral theory, we know that kangaroo rats know their neighbors. So even though they're solitary, Jojo knows that Lily lives over here, and Wiley lives back there, and Claire lives over there, and Zoe lives over there. They know their neighbors, right? And they're more likely to mate with their neighbors than they are with strangers. So I thought, well, what if they're actually dependent on knowing where their neighbors are, and if they get moved without them, it's a problem. So I went about and we moved 100 animals. We pulled them off of areas where they were going to be either plowed under, over here, or MWD wanted to do some conversion. This is Metropolitan Water District, wanted to do some habitat conversion, and they were being poisoned in the parking lot. So we pulled them off those areas, and we put them onto the reserve in groups that were either with their family or with their neighbors that they knew, or without their neighbors that they knew. And we found that it worked. So the blue bars show that if you translocate kangaroo rats with their neighbors, they're more likely to survive at 1 month, at 3 months, at 6 months, and all the way up to a year after you've translocated them. So it was super exciting. But what the kicker was that not only were they more likely to survive, but females that were moved with their neighbors were significantly more likely to have babies. So the ones that were moved with their neighbors had 24 times more babies than the ones that were moved without their neighbors. So it had a huge effect. In fact, this would determine whether the translocation was successful or not. And they were more likely to settle on the site where we put them when we moved them with their neighbors. So this is how far they moved before they settled. And you can see that the ones that were moved without their neighbors moved a lot farther than the ones that were moved with their neighbors. And it took them a lot longer to settle, because this is days to settlement. It took them a lot longer to settle than it did the ones that were translocated with their neighbors. I'm just going to skip that one. And you might be wondering, what about their behavior when they were released? I did say I like to study behavior? Well, the ones that were released without their neighbors fought a whole lot more when they were first released. But the ones that were released with their neighbors spent a whole lot more time eating right out of the acclimation cages, right when we released them, and digging burrows, which was really exciting because that's what a kangaroo rat should be doing. And over the long haul, we looked at it for 5 years after we released them. They actually shot up so high that we had, this is what we consider to be high density, is about 8 per hectare. We got up to actually 35 per hectare in the first year, and it sort of balanced out around almost 16 per hectare. So we did really, really, really well. So the next question I wanted to look at is, could we find a way to dampen dispersal? And the kangaroo rats, like I said, they use sand bathing to talk to each other and to drum their feet to talk to each other. But in this case, we thought, well, if we're trying to get kangaroo rats to stay on a release site, maybe if it smells like a kangaroo rat when they get there, they'll be more likely to stay. So we tested this by putting kangaroo rat scent around our release cages on for half of the kangaroo rats, but not for the other half. And we found that sure enough, if you release them with scent from other kangaroo rats in the area, they're more likely to stay on that site than if you release them without the scent. There were survival differences too, so if they're with kangaroo rat scent on the release site, they are more likely to survive at the 1 month mark, but by the 3 month mark, the whole site stinks of kangaroo rats. So at that point, there's no effect of moving them with the scent on survival. So it's a short-term effect, but it still helps in those first risky weeks. So as I said, pretty much everything wants to eat the kangaroo rats. Everything out there. And so we go about putting them in these individual cages, because each one they're solitary. If you put them in together, you're in trouble. They might hurt each other. So we put them in these individual cages, and we put an electric fence around the site to try to keep the coyotes off. And maybe we could keep the skunks off, and maybe we can keep the raccoons off. But turns out that they get on there anyway. So I sort of reasoned from behavioral ecology that, well, maybe what's happening in our landscape is that because we've gotten rid of so many of the mountain lions, there's a higher number of coyotes on the site. So we see a mesopredator release is what it's called. So there's a lot more coyotes and far fewer mountain lions. That's a problem when you're trying to release a prey species, when that predator eats the prey. So I thought, well, what if, mountain lions are in this area, right? So what if we put mountain lion scent on the site to see if we could keep the coyotes off? And if we kept the coyotes off, maybe we would do better with the kangaroo rat release. So we looked at that. We, again, pulled animals from a couple of different sites, and we moved about 150 of them into 6 different areas. Half of them we seeded with cougar scent, and half of them we seeded without cougar scent. Where? Yeah, Zoe.
If we put the mountain lion scent on, and if the scent works, then won't the mountain lions try to get inside and get electric shock?
There isn't. On this side, I didn't put the electric fence up. I was just using the scent. And so people often ask me, where do you get cougar scent? So I want to use a strategy that other managers are going to be able to use. Did you know you can buy cougar pee online? You can. PredatorPea.com or Leg Up Enterprises sells cougar pee. And I bought it from there rather than collecting it from the cougars at the zoo because I wanted it to be a strategy that would work for other managers. So I did that. So here's what we do. We go through the process of basically prepping the site. We have to remove the non-native grasses. In this case, we had Cal Fire come out and do the burn for us. Then we put in a below-ground cage and some artificial burrows, and we go out there and trap the animals that we want to get and in order to move them. And then we hold them for a period of time. we hold them in this acclimation cage where they can go up and down between the lower part of the burrow below ground and the top burrow above ground. And then I go out there and I spread a bunch of urine around on some of the sites, but water around on the other sites. And then we go back out there and we try to figure out who's there and how well they did. So on this study, we actually put remote cameras around the entire sites, all of them, so that we could document the visitation by the coyotes and the skunks and the raccoons and such. What we found was that it worked. So if you put cougar scent on the sites, you have a higher amount of kangaroo rat survival at the different time points. And the kangaroo rats, let's see, this is a burrow establishment, and they are establishing more of their burrows on the site with the cougar scent than on the site with the water. So they're getting their burrows in. We had so many pictures of predators out there. Pretty much everything you can imagine was out there. Right, you can see the top cage for the kangaroo rat, and there's a skunk right there. They were all over the place. We had a lot of coyotes, though. Just a ton of coyotes on all these sites. And I got some video that I want to show you. So this is a scented site, so I was pretty optimistic about what was going on here. The coyote does not like the cougar urine, right? So I was really hopeful that it was really because the differences were because it kept it off. This is an unscented site. The coyote knows something's there, but it's not super worried about it. So what I was predicting was that the cougar urine was going to keep the coyotes off, and that's why the kangaroo rats were going to do so well. But that's not what happened. So the cougar urine really didn't have an effect on the visitation by all of the predators. I counted up all the predators that I saw. But when you separated the predators, you took those bobcats and coyotes out and separated the size, the large predators, from the smaller predators, the foxes, the raccoons, the squirrel, the skunks, and the weasels. It turns out that it dampens the visitation of the smaller predators, and those are the ones that are probably taking the kangaroo rats. So, in the process of doing this project, I was tasked with coming up with a model for translocation. And I went through the process of asking multiple questions and ultimately coming up with a summary of recommendations, which are extensive. I also, part of the work is not just doing the research itself, but it's actually communicating that. So luckily at the zoo, we have a fairly active PR department. And so we're able to, publish papers, but also get information out to the layperson about how effective these kinds of strategies are. And it takes a village to do this kind of work. It really took a lot of effort. We did work with CAL FIRE, like I said, and we work with Metropolitan Water District, and we work with the California Department of Fish and Wildlife and the U.S. Fish and Wildlife Service. So it really took a collaborative effort to make this work. But we've designed a program now for the kangaroo rats, and we have effectively established 6 new populations. We've moved thousands of animals, and I'm happy to say that they're thriving on this reserve. So as long as the reserve managers continue to manage that habitat so that it keeps it open, we have a strategy now to do this effort. So I wanted to switch gears and talk a little bit about a smaller species. This is the Pacific pocket mouse, which is also a heteromyid that's found here in Southern California. And I want to ask the kids, it's super tiny. I want to ask you, who thinks it weighs the same as 1 penny? Who thinks it weighs the same as 2 pennies? Claire. Who thinks it weighs the same as 3 pennies? Perry and Wiley and Gabriel and Sarah. Or 5, yes, well, you guys are right. It weighs the same as only 3 pennies. This species is between 5 and 7 grams. It is the smallest mouse in North America, and we have it here in California. it lives in the California floristic province, right? We have this amazing biodiversity hotspot along the coast in California. Unfortunately, that's also where we've developed. So there's this conflict between wildlife and humans. And this is one of the species that's been placed at risk. So historically, there were several populations from LAX, so the El Segundo Dunes, down to the border with Mexico. Back in the 1980s, though, it was thought to be extinct. And it was rediscovered in 1993 at the Dana Point Headlands, and emergency listed as endangered by the federal government. Currently, well, I should say, as of last year, we had only 3 populations left, 2 on Camp Pendleton and 1 at the Dana Point Headlands. And no new populations had been discovered since 1995. I stuck this little guy out here because we use scent dogs as part of our program to try to find the pocket mouse throughout the habitat, because there's a lot of habitat to cover. And we basically use that we can train them on the pocket mouse feces, and then they can go and direct our trapping efforts by telling us where they think they've smelled them, which is really interesting. Pocket mice are found mostly in coastal dune scrub habitat between the coast and only 4 kilometers in from the coast. So they're restricted to a band of habitat that is highly developed. There's not much, if you think about it, there's not much habitat between LA and the border that's not developed, that's right along that coastline. So they have mostly, a lot like many other species, the threats include development or habitat fragmentation. And because we've driven them down, they have very small population sizes. We also have some impacts by the military training, which the base is, Camp Pendleton is very careful to try to keep the training outside of the critical habitat for the mice, but sometimes they need to use those areas, and just walking in the habitat can compact the soil and actually crush the mice because they burrow only between 1 inch and 10 inches underground. Fire is also a huge risk on Camp Pendleton. This is a shot from the Dana Point Harbor, and we have a lot of fire. And with only 3 populations remaining, we have a need to put more animals out there so that a catastrophic event won't wipe out the remaining couple of populations. So we've done a bunch of research in the wild. We looked at home range size and when they emerge and sandbathing. I'm going to show you, though, just a little. So I wanted to tell you a little bit about this, and then I'm going to show you a video. So we track pocket mice, which is really hard to do because they are this big. And to do that, I found a butterfly biologist in Switzerland who designed a tiny little transmitter for the pocket mice. And I put it on candy necklace cord. Do you guys know what that is? Yeah, it's stretchy. Yeah, well, I made little harnesses for them. See those little loops? I put those around their arms, and I managed to get this thing on this kangaroo rat, and I can track it around the habitat to figure out how far they move and who they visit and what kind of habitat they like and all sorts of things like that. And that helps us because we can figure out where we want to put them when we put them back out into the wild. And this is basically what we get. We get like, this is how big their home ranges are, and this is 10 meters. So this room would be big enough for like 4 of them because they're only this big. They don't move that far. Okay, so what we know, I'm just going to tell you a little bit about what we know about them, and then I'm going to turn on this video. So males come above ground in February. Females come above ground a couple of weeks later. They mate. They have about 1 or 2 litters. They can have 3. And then males start going down, and they go through what's called estivation. It's different than hibernation. because in our dry adapted habitats, it doesn't get really cold along the coast. And that's the trigger for hibernation, it's the cold weather. But estivation is also a period of inactivity. It's just that it's triggered by low food availability. There's just not that much food. And so once in the fall, they detect that there's not that much seed out there, they start going down. So males go down, and then females go down, and then they stay down from September to February. really showed you guys what it took to get there, which was a lot of work in the captivity. These guys are like a tiny panda. Because they're solitary, they are just as likely to try to kill whoever you put them in with than they are to mate, as they are to mate with them. And so it's really, really hard to get them to breed. And so we got to that relocation last year. The first reintroduction was truly incredible, but it took us 4 years to get there, and it was learning about how to breed them. We think of mice and rats as something that just breeds like crazy. This species has 1 to 3 litters a year, but they only come into peak estrus for about 1 hour every 14 days. And if you don't get the right male with that female in that hour, and it could be at 3 in the morning, right? You try 8 different males, and she doesn't want to pick him, then you've missed an opportunity. So it took us 4 years to grow from 30 animals to 110, but we did it. And that's why we had enough animals to do the relocation, the reintroduction last year. Since that time, we have just recently gone back out and put another 25 out onto that site because we want to keep supplementing until we have a viable population. They are actually currently in acclimation right now, and they're going to be released if it doesn't rain on Monday. So fingers crossed that it goes as well as we hope. If they do well, we will have established the 4th population, and we'll go from there. But I also work with the Southern Mountain Yellow-Legged Frog program. I have a captive breeding reintroduction program for this species. This is a species that it lives in, it's diurnal, and so it's completely different than the pocket mice, right? They're about 3 inches or so, and they actually vocalize, but not like most frogs do. They vocalize underwater. Clicking is their vocalization. That's what they use to coordinate mating. Oh, let's see if I can turn that off. There we go. And they're found throughout the San Gabriel Mountains, the San Bernardino Mountains, and one site is in San Jacinto Mountains. And so we're working up in the San Bernardino National Forest with this species. They have a lot of threats that are different threats than we see with the heteromyids. We have pollution in the streams, obviously, and there's worldwide amphibian decline that we're dealing with, and so UV radiation is a huge problem. All of our local lakes have been stocked with trout for fishing purposes, so for human recreation. And the trout breach the dams. They breach the dams and they get into the local creeks, and they eat all of the frog eggs. And so that's one of the main ones. We also have introduced bullfrogs out there. And most recently, the biggest issue is chytrid fungus. So we have fungus that has been pervasive through our stream systems, and as these populations decline, then the chytrid effects take over. So we basically brought into captivity 86 tadpoles back in 2006, and we set them up in tanks, and then we grow them up to breed them and then release them. And we do. For these species, we're dealing with water quality issues in captivity and diet. One of the things I wanted to just say really quickly is that while for the kangaroo rats and the pocket mice, the first thing that we looked at was behavior to improve release success. For these guys, they live in high elevation streams. And so what we found, we decided to basically refrigerate them in captivity because the first 2 years we weren't seeing any reproduction at all. And so when we refrigerated them for 3 months, we found that it had a huge impact on their behavior. So males were more likely to call and exhibit reproductive behavior than they were when we didn't hibernate them. And we're also looking at mate choice and a bunch of other things. But this is also a super cool little video that I wanted to show you. It's a frog egg turning into a tadpole. And what you can see is just the process of development. We took this with a GoPro and we've sped it up for you. And what you'll see is it developing. Can you believe that's going to be a frog? So, so basically they go through that development, they become tadpoles, little tiny tadpoles, then they get bigger and bigger, and then they sprout some hind legs, and then they get their front legs, and then they start to reabsorb their tail and become little frogs. Look at these numbers compared to the mice. So we're dealing with a completely different animal. We're getting 6,000 eggs, right? Compared to where, we might on a really good year with the mice get 25 or 30. We're dealing with 6,000 eggs, which turn into 2,000 embryos, so we lose a lot along the way. This was 2015. Again, we had 6,000 eggs in 2016 and about 2,000 embryos. So we've done really, really well with the frogs, and it has such amazing reproduction potential that it's really easy to put a lot of animals back out into the wild. And look at the difference here. Basically, we can take the animals we produce in captivity, and we can transport them in trucks in a bucket, and we can dump them into the creek. It's a very different process, right? We have found that if we release the juveniles, so if we head start them for a whole year, they're more likely to survive the release than if we release the tadpoles. But we're just starting to learn about their behavior because technology has improved so that our tags that we can put in them are small enough that we can mark our juveniles. And now we can start asking some really interesting questions. We go out there, we survey after we do our reintroductions. We actually swab them because we're trying to see how long our treatments to keep the chytrid off of them lasts. So we swab them every time we go back out there. And we're looking at how far they move, just like with the pocket mice. And it turns out that these are the little squares are where we release them, and the circles are where we find them. So they're moving really far, and they're more likely, they're moving really, really far. One of them actually moved 709 meters. So when you release them, they really do move a long distance, or they can, and they're more likely to move upstream than they are downstream is what we're starting to learn. And remember, I told you that they were more likely, the hibernated ones, the males did really well in terms of their reproductive behavior. Well, it turns out when you hibernate those metamorphs, the juveniles, they are more likely to survive a release, and we don't yet know why. So there's so many more questions for us to ask. There's so many species that need our help. But we're making headway, and again, it just takes a huge collaborative effort. So I always like to end with this slide. It's my favorite quote because most of the species I study are not rhinos or elephants or sharks. They are small. I consider them to be charismatic mini fauna. This is a quote by Aldo Leopold. "To keep every cog in the wheel is the first caution of an intelligent tinkering." And so when we fragment and destroy habitats, it's really easy to forget about the little guys that are out there. But they can be extremely critical for ecosystem processes. So now with that, I'll take questions. Does anybody have any questions for me? Lily, did you have a question?
Yeah, there was a map and there was a star on it.
I'll go back. You can tell me which one you're talking about. I think it's.
Yeah, it was the star that we found near the.
It might have been where we released them.
There.
Yes.
Yeah, Riverside. It wasn't on the key.
Yeah, it's not in the key. You know why? It's because I think the key is cut off on here. It's because it's not cut off. Good question. I love that. That is our first release site. That's where we released the frogs the first time. Yeah, good question. Anybody else? Jojo.
When you were saying like every time you go back you swab them, wait, why?
So they get, because they're out in water in our creeks, and there's a fungus out there in the creeks too, the fungus gets on their skin and it makes them really, really sick. So before we release the frogs, we have to bathe them in an antifungal solution to try to give them a little resistance to that fungus. And so we do that, and then we release them, and every time we catch them, we go out there and we swab their mouth, and we swab their back and their bellies, and then we turn those in and we run them in the lab, and then we come back and we find out, do they have chytrid yet? So we're trying to see how long we can keep them fungus-free. Thank you. Yeah.
What happens to your job when the
Good question.
when the governmental budget is constricted?
Yes.
And how do you convince the surrounding area or the human population that this is a very, very important project that you're doing?
Good questions. What about my job? Well, I'm fortunate to work for the San Diego Zoo. We have an amazing membership down there. We have incredible development department and phenomenal donors. So a lot of this is supported by the government dollars, but a lot of it is supported by private citizens. So I think my job is safe. Luckily, I have an endowed position, so I'm okay for a while. But it is true that we are all very nervous about what's going to happen with conserving species because ESA is being threatened. So that's the first part. The second part of your question is, how do you convince people in the area? Well, that is really hard because sometimes we can have multi-use areas. So we just designate habitat as a reserve, and then that's where basically we preserve the animals. Everywhere else, we assume they're going to be gone, right? They're going to be extirpated from all other lands. And in those reserves, if they're multi-use areas, well, humans are using those too. So for the frogs in particular, it can be really tricky because they live in creeks that go through campgrounds. And when people are in the campgrounds, they want to be in the creek. And they kill the frogs. So we have, in some areas, we designate critical habitat and we don't let people into those little areas. And then we find other areas that people can use. Because we want to make sure that there's a balance, that people can use the parks, but also there are areas for the wildlife. Yeah.
Thank you.
Yeah.
In the area you mentioned that the pocket mice were thought to be extinct, and maybe I might have missed something, but how do you identify that they're there, and how do you collect the mice? I mean, you put those little traps.
Yeah, so
that they're there and put traps.
Yeah, we do. So before we started using the scent dogs, we basically look for habitat that we know they prefer. And then we go out there and trap those areas. And we put out those Sherman traps, those little box traps that are live traps, and we bait them with seed. And we trap usually for about 5 nights in a row, and we look at, there are other small rodents in the areas where these guys live, and so we might catch spiny pocket mice, we might catch deer mice, we might catch harvest mice, Rethrodonomys megalotus, but they're distinguished, by these guys are the smallest species in the group that they live in the group of sympatric rodents, and so basically what we do, and I sort of rushed because I wanted to show you guys that video, but what we do for collections is we trap an area. So this is the Dana Point headlands, and we basically put traps over the whole thing, and then we got really lucky with this one. Actually, it was really easy because we caught 26 animals through the whole site, and we wanted to collect 10. The goal was to collect 10 from each of the source populations. And so we got 26, and I was actually able to pick and choose which individuals I wanted to bring in because I wanted an equal sex ratio, and I wanted to have some adults and some young of the year, so I knew that they weren't really old because we can't age the adults. So they could be 3 and still be out there, and I didn't want to bring in really older animals. So we were able to basically go out there, and we use traps, we collect them, and then we transport them to the facility. Julie.
Do the little teeny tiny transmitters affect their behavior?
We have not seen any effect of the transmitters. Some animals just don't tolerate the transmitters. I did put them on the kangaroo rats as well. And if you put them on and they, can't move their arms properly, or they seem to be freaking out, because some of them do, we just take them off and we'll find somebody else that can tolerate them. Zoe?
I have 2 questions actually. The first one is if we make the fences not electrical for the mountain lions, still would they be able to tear it down or jump over it?
They can jump over it so easily. It's only this tall, and coyotes can jump over that too.
Yeah.
Second question. Do you have another question?
At the very front, there was something on a kangaroo's ear.
Yeah.
Kangaroo rat.
Yeah, you know what that was? That was an ear tag. So when I go out there and I watch them at night with my night vision goggles, I want to know who I'm looking at from a distance. And so the way I do that is I put a little ear tag. It's like an earring in their ear, but I put reflective sheeting on it. So, sometimes when you're driving around, you see signs that are reflective at night. Well, they make those in all colors, and they're like little stickers. So I can put little stickers on their ears, right, on their earrings. And so I can look from here all the way over there, and I can see that's blue-green, and I know that that female lives there. And that's green-yellow, and I know that that male is going to go visit her because I'm watching. Yeah. Yeah.
Do you have those kangaroo mice in Topanga?
We have kangaroo rats here. We do. The Dulzura kangaroo rat is here. They are the Pacific kangaroo rat. And there are 19 species of kangaroo rats in total, and all are on the decline.
So they are on the decline in Topanga too?
Oh, yeah. They are considered to be a species of special concern in California, the Dulzura kangaroo rat, the one that we have here.
We've seen them 2 different times. One time during the day at the Topanga State Park.
Yep.
And another time during the day at an Easter egg hunt recently.
Oh,
And we assumed that they shouldn't be around.
they shouldn't. They should be asleep.
Something that we should have done. Okay.
Leave them alone.
That's what we
Most
did.
likely, if they're above ground, there's something wrong.
Yeah, that's what we thought.
Yeah.
Okay.
Yeah.
So they have the cute little
Backpacks.
transmitters, but the thing that you put in the base of the tail, what is that?
That's
And
called
does
a-
it do something different?
It does. So we marked the kangaroo rats with the ear tags, the little earrings that you talked about. But the earrings, the smallest ones that they make, are way too big for the pocket mice. One of the challenges when I started working with the mice is that they're so tiny that every possible method we have ever used for any other species of mammal does not work for them. Nothing worked. So we had to completely rethink how we did basically all of our work. And so we started, actually, with something that Greg suggested to me when I first started the program, was to use a visible implant elastomer, which is a silicone-based elastomer that you color, and you can use a black light and shine it on their tail. And so I would create color codes in their tail. But there's a limited number of codes that we could use. And so 2 years ago, the pharmaceutical industry designed these new P-chips, and they did because they needed to mark their lab rodents. So it was really designed for lab studies, but I took them and put them in the mice. They are the size of, you could see them, they are super tiny, you can barely see them. And because of that, we've managed to be able to track individuals following release. We tried them with frogs, but they're too liquid, they're aqueous, right? So when you put them in, the tags flip over and then you can't read them. So the pharmaceutical industry, the Pharmaceutics that designed them are actually designing new tags that are readable on both sides now so that we can use them for more wildlife species. Lily.
Why did you choose to show both instead of one?
Both species?
Yeah.
We did really different things with the two. Well, I showed you 3 different species, right? The kangaroo rats and the pocket mice and then the frogs. I just wanted to show some differences between what we were able to do. The kangaroo rats, there were enough of them that what we needed to learn was different. We could do some research and study them in the wild and do translocations where we move them from the wild to the wild, but we could study their behavior. But the pocket mice, there weren't enough of them. There weren't. I went out there in 2006 and there weren't enough of them for me to do that. So I had to bring them into captivity so I could grow up a whole bunch of mice before I could create a new population. Jojo.
What's their lifespan?
The kangaroo rats or the pocket mice? In the wild, a 3-year-old is pretty old. In captivity, probably around 6. Jojo.
Are the kangaroo rats like called kangaroo rats because like do they have like kangaroo legs?
Yes, you got it. That's exactly why. They're bipedal. They stand up on their back legs. Now, I could do it for you, but it might be a little embarrassing. They stand up like this. They have tiny little front feet and really big back feet, and they hop. And they have a really long tail, and their really long tail helps them stay balanced. Exactly. Exactly. And so when a snake strikes at them, they can ricochet out sideways 3 feet like that. It's pretty amazing. Yeah.
It's amazing.
Yeah. Well, thank you, everyone. Oh, you have more questions. Okay. Zoe.
There are 2 videos on when the coyote went the scent and the no scent. Was this a mountain lion?
Yes.
Oh.
Yeah. And they're scared of it. Did you see that? The camera had tipped sideways, but yeah. Lily.
I forgot.
Okay. Jojo.
Would it work on like other species of predators if you use scents from the predators of those animals? Would it work to scare those ones away?
On other it might. I don't know. We'd have to test it. Julie, did you want to ask me a question?
Just one little one. So because their sense of smell is so highly developed, do you think that they know you by your scent and they're less afraid of the researchers because they know you, or because it seems like they weren't running away from you?
They don't. Well, you have to remember that when we're out there and we have our headlamps on, they're blinded.
Okay, so that's what makes them.
Because they're nocturnal. So if you have them in your headlamp, they can't see anything.
I see.
So they're not going to run away because they'll run into your leg.
Okay.
Yeah.
Thank you.
How small was the smallest kangaroo rat?
There are lots of species, but one of them that I study is like that big. Think about that. The one that the one that I showed you is more like that big. But there's one in the Central Valley of California called the giant kangaroo rat. He's like that big. Not as big as a Tasmanian devil, though. They're like that big.
Doctor, do you have favorites that you miss when you let them go?
Oh,
Is it
sure.
sad?
It is sad. Last year, I had a male who, he wouldn't go down in his acclimation cage burrow. He wouldn't go down. I stayed with him all night, and he wouldn't go down. And in the morning, I took him out of his cage, and I opened up the other burrow, and I put him in there, right? And I hoped that he would, nope, I came back an hour later and he was up in his cage again. So I took him home. I didn't take him to our home. I took him back to the captive breeding facility and I got a different male and I swapped him out. So certainly it's hard to see that. And we don't want to release animals that won't make it out there. So we really do everything we can to make sure that they have the appropriate survival skills. I talked about anti-predator behavior in the video. We also look at foraging behavior and shelter use behavior. We're actually right now, we're just starting to look at shelter use behavior. So we're actually taking water and putting a little spray of water on their sand in captivity. And for all of the ones that are going to go out, we look to see what kind of a burrow they build, and we'll foam those burrows. So we'll put them into wet sand. They dig a burrow. We trap them out of that area, and then we use foam, and we foam their burrow so we can see the structure of the burrow that they're building and make sure that they have nest chambers and that kind of thing. Kent?
So with the relocations and the reintroductions, when you have one has poor survival or worse than you prefer, is there a primary reason for failure? Is it predation? Is it old age? Is it failure to mate? Is it infection? Or is that different depending on where you're trying to put them?
Exactly. So we don't often know what the mechanism is, and that's what we're trying to get at. So often it depends on the species, right? So for the kangaroo rats and the pocket mice, it's predation. And if we release the tadpoles for the frogs, it's predation. There are corvids that come in and pick off the tadpoles like they're, snacks. But for the Tasmanian devils, when we do the releases, it's cars. So we lose 30% of the Tasmanian devils that we release to car strikes in the first 2 weeks. So every species, we're trying to do something different. For the devils, we're trying to train them to avoid cars. For the pocket mice, we're trying to train them to be savvy with predators. And
is the frogs the one that's sort of unusual in terms of it being infection?
for frogs, yeah, because the chytrid is prevalent throughout their system. There's just no way we're going to be releasing them into a disease-free area anymore. Lily.
You said something about you were gonna release more if it didn't rain on Monday. What does the rain have to do with it?
So they're in the acclimation cages right now, and if we take the cages off and the top cages off and it's raining, I just don't know if they've never been in the wild before. The first thing they're going to have to do is what?
Dig a burrow?
Probably. Now they can use their acclimation cage, which is, a foot underground, so it's pretty safe and cozy down there. But if they come above ground and it's raining and they get confused and they start running around and they try to dig a burrow, it's not going to be a really good burrow in the first day, right? It's going to take them a few days. So it's probably safer for them to stay in the acclimation cages than it is for them to be released while it's raining. Jojo.
Do you work with like other animals like predators?
Yeah, so I work with the Tasmanian devil in particular, and that species is a predator and a scavenger. So the cool thing about them is when you release them, you don't have to worry about them finding food because there are roadkill all over the place that they go and eat. Yes.
Did you know that there was a species called the Tasmanian tiger, but it went extinct?
I do know that. Yeah. Yeah. People keep thinking that they see one. Here and there, every couple of years, there'll be a sighting, but we haven't verified it yet. Lily?
Do you know that Tasmanian devil's jaw is so strong that it can break open a bone to
I
get
know.
at the marrow?
Yeah, it's pretty amazing. Yeah. Jojo, one last question.
Do you have a favorite species that you study?
That's a good question. It's hard. I really, truly love them all. But, maybe the kangaroo rats. Maybe them. Although the prairie dogs are, every species I've ever studied is interesting in their own way. And the prairie dogs are really fun, which I didn't talk about here either, because they live in their family groups and they're really social. And if you get to know a baby and you bring it into captivity because, say, its mom is hurt or something, they want to cuddle with you, so they're very sweet. Well, thank you so much, everyone.